let's begin cellular respiration. So, to begin, glucose enters our cell. So, the way that works is glucose formed from eating food and digesting it diffuses into our bloodstream, or for a plant cell, it is created from photosynthesis, and oxygen, which diffuse into our body cells from your bloodstream. It is then used to be broken down to get into the mitochondria, which powers the cell. Because cellular respiration is basically the process of breaking down energy and fuel to create ATP. So, once our glucose is in the cell, glycolysis can then begin. Glycolysis is the process of breaking down glucose and oxygen to get into the mitochondria to power the cell, and it occurs outside of the mitochondria in the cytoplasm. So, the glucose, now on the outside, can then be transformed. So, the cell uses two ATP molecules of energy to separate the bonds of glucose into two parts called pyruvates, which is right here, called pyruvates. These pyruvates are located on the outside of the mitochondria so far and are used to be stripped of their electrons and hydrogen atoms by NAD and NAD+. So, NAD is an electron carrier located in the cytoplasm outside of the mitochondria, and its job is to strip the carbon molecules of their electrons and hydrogen atoms, and that causes the NAD to become NADH, which is this. NADH, once having collected those hydrogen atoms and electrons, transfer these electrons to other reactions happening in the mitochondria. So it basically happens whenever electrons are stripped and stored from NAD to create NADH. So it's beginning to travel with these electrons. And then only 2 net ATP is produced from the pyruvic acid, because now the pyruvate has now become pyruvic acid because NADH has taken its hydrogen atoms and electrons. So only 2 net ATP is produced from the pyruvic acid molecules after glycolysis. So not that much energy. So as the pyruvic acid goes into the mitochondria, once carbon is removed, creating carbon as a byproduct. So coenzyme A. So now that you have that, the little pyruvic acid is now ready to move into the mitochondria. So you can take this away. And it's now ready to move into the mitochondria. So now it's in, okay? So this begins, and then coenzyme A comes and attaches to the 2 carbon fragment and is now ready for the Krebs cycle. So, during the Krebs cycle, located in the mitochondria, before the cristae happens after coenzyme A, and helps to break down the last little bit of sugar of the 2 carbon skeleton of glucose, is attached to an existing, so this little 2 carbon, we'll now say this little 2 carbon molecule of glucose, excuse me, this little 2 carbon molecule of glucose is then attached to a 4 carbon molecule to create a 6 carbon molecule. The new 6 carbon chain is broken down, releasing carbon dioxide and electron carriers again, and electron carriers again come to collect the electrons from being broken down. So now you have 2 ATP molecules are produced for each molecule of glucose that is broken down and all that. So now we're left with this 4 carbon molecule that has created ATP, ATP molecules from this whole Krebs cycle. And now you're at a point where the NADH has come and kept taking the electrons from this, from the molecule being broken down over and over again, another coenzyme coming and adding while the electron carriers collect enzymes, starting the process over again. Well, that's happening. FADH2 is formed. And this happens through the process where it comes in, because it's already located in the mitochondria, and it comes in and takes electrons from the 4 carbon compound, which is happening right now, from the 4 carbon compound, and leaves, so it comes in and takes electrons from this 4 carbon compound and leaves, making the 4 carbon compound unenergized. And that way, the electron carrier coming back in from glycolysis and just keeps going around in a circle because it keeps adding electrons, and then so it keeps going in a cycle, if you can see. So anyway, this FADH2 then comes and transfers and travels, and the NADH is over here too. So FADH2 is now coming, and all these electron carriers and transfers. And so now we've hit the final stage where we get the electron transport chain, which is here. Okay. And so now, after the Krebs cycle, you were left with a 6-carbon compound, and the NADH that you got from the carbon compound loses its electrons, and so they separate and become NAD, and they leave. And so this leaves again, and now this just comes out, okay? Or it's been done, so it goes back and it goes and gets more electrons. See, so that's a never-ending cycle. So it goes back to get more electrons, and then, so now it's become NAD, and they leave, and then they come back. So the hydrogen electrons are on the electron, and they leave the electrons here on the electron transport chain. And they go to the proteins on the crystalline wall, which is in the inner membrane of the crystalline wall. And you are now here. Of the proteins on the crystalline wall that pushes them towards the other proteins on this electron transport chain. And the third protein adds oxygen, and it forms water as a byproduct. So, and also CO2, which is why we exhale CO2 as a result from respiration. So that is also out. Okay, and then the hydrogen pump, which is now here, is powered by the electrons flowing through the electron transport chain, which powers the pump to push the hydrogen ions out through ATP, and thus creates a net amount of 80, pushing it out, creating ATP, which creates a net amount of 32 ATP molecules. So, meanwhile, all of this, I forgot to mention, was part of the matrix. And the matrix is located inside of the mitochondria and is used to harbor a spot for all these cellular reactions to occur. Along with the cytoplasm, which is out here, where glycosis originally began. And that is located on the inside of the cell and is kind of like, so now we're finally left with ATP. 32 net ATP molecules have been created, which now creates energy for the rest of the cell to function and to basically give energy to our bodies. And that is why they call the mitochondria the powerhouse of the cell.